Thank you for coming. Now we are going to, I'm going to start now about exciting talk about something you have all been waiting for: bears, bear poop, and DNA analysis. Whenever we talk about this to other people, everybody laughs and giggles because who doesn't like poop? So I'm going to tell you what we do here, where I come from and where I sit as a researcher. Just quickly, I'm all the way up here in the north. You can see the Arctic cycle here. So currently we have midnight sun. And there's another picture from where I live. Despite being so high in the north, you can see it's quite green. And currently I'm sitting somewhere here in this house talking to all of you. So something about just a quick introduction to what I'm going to talk about today. I'm going to talk a little bit about brown bear history and management in Norway and Sweden. Yes, thank you. It's a very nice place. The monitoring, the available methods, and what we do here at the moment. And then how does it work and what can we use the data for? So, but first, to kick things off, a little quiz, and you can give the answer in the chat. So you are able to solve this. I give you some information. Bears have a very short intestinal tract, but they eat everything. They are omnivores. And because the food spends so little time in their stomachs, foodstuff, so the things that they eat, are not easily digested and broken down. So very often you can see very well in the fecal matter what things the bear has been eating. So if you see these four pictures here with that information I just gave you, what do you think is the bear poop? And you can give your answer A, B, C, or D in the chat. I will wait a little bit and see how many of you get the right answer. It's of course also the size that matters. Answers are coming in. Come on, somebody else venturing to give me an answer? Yeah, very nice. We do this test always. We have sometimes children here and they get a diploma afterwards. Bear poop experts, they love that. I don't have, unfortunately, I cannot give you all a button. Yes. I'm not exactly sure how many have signed in. So I have 1, 2, 3, 4, 5, 6, 7, 8 answers now. Yeah. Okay, one more. Is one more going to answer? Otherwise, I'm going to reveal the. Yeah. Otherwise, I'm going to reveal the answer now. Okay, everyone ready for the answer? I will go over to the next slide. So here's the answer. The first poop was from a. Yeah, I see somebody is not happy with the general answer. No, yeah, you got this right. C, exactly. So here, this is the poop from the bear. This D was from a hare. B was from a fox. It's often, you know, like this, a little pearl string. And A was from a moose. If I go back quickly, you can see that there are some berries here in the poop. And that you can often see when we get fecal samples here in the lab, that we can tell exactly. We know if the bear has been eating ants or grass or blueberries or lingonberries. So that's very nice always to be able to see that. It also smells like the stuff it has eaten. So that's sometimes it's nice, sometimes it's not so nice. If it has eaten meat, for example, the poop tends to smell quite badly. And berries is always nice. We love the blueberry poop is the best. That smells like blueberry and it doesn't stink up our lab. Okay, so after this we go further. A little bit about brown bear management. So here on this map you can see all the brown bears that currently exist in Europe. This map is a little bit dated, but I didn't find a newer one. You can see most of the brown bears exist in Northern Europe here. Sweden, Norway, Finland, and Russia. And in Russia we have the largest population in Europe, the largest number of bears, which is about 40,000. In Finland we have about 2,000 bears. In Sweden we have about 2,700. In Norway we have not estimated the population size. I'm going to tell you a little bit more about this at the end. But we know there are at least 136 bears in Norway. And then I have gotten some more information, but not with, with the sighting like this here, that in Romania we have about 6,000 bears. And in Greece, down here, we have about 400 bears. I don't know about the other countries. But that's it's not too bad. It was once better. The brown bear has lived all across Europe, and unfortunately it does not anymore. But the populations are on the way up, so that's very nice. So I'm concentrating in this now on Sweden and Norway because this has been researched extensively, and that is the stuff I know. So this is the demographic population history. Demography is how the number of the bears develops itself. And you can see here the yellow one is Sweden and the blue one is Norway. This is the timeline, so 1900, 1950, 2011, and this is here on this axis here is the number of bears. And you can see Norway started out with more bears than Sweden. Over 3,000, while Sweden had only about half as much as Norway. But then in the beginning of the last century, both countries decided they wanted to extirpate, so get rid of all the brown bears in their countries. And the population, the numbers went quickly down. Norway was very effective in killing all of its brown bears. And then around the 1950s, people started to think that, no, maybe it would be nice to have some bears in the country after all. And Sweden was first with deciding that they wanted to stop decimating the brown bears. So it stopped here with some bears left in the country. But Norway only abolished paying money for hunting brown bears. So Norway was quite successful in getting rid of all the brown bears in their country. And that is why in the 1970s, when the brown bears became protected, also in Norway, the population number, so the number of bears, only rose very slowly, while as in Sweden, you can see that this number goes up very quickly. And then in 2006, in Norway, was monitoring introduced, so that you track all of the development of what the brown bears are doing in the country. So you have a bit better control about what is happening. And here you can see another way of displaying this. So this is the beginning, 1860 to 1870. The gray shaded area here marks where there are no permanent bears living there anymore. So sometimes you can get a visitor, but they are not there all the time. And you can see as they progress in killing the bears that this gray area creeps up north over, and then by the 1920s, almost all of Norway, there was probably one visiting there here, all of Norway is empty for bears. And in Sweden there were some bears left here in this area. So I have on the Polarpedia, I'm giving some information about the Berne Convention. That was introduced or was in force since 1986, and it is a legally binding treaty that is aimed to conserve wild flora and fauna, so all the plant and animal species, and their habitat, so where they live. And this treaty is international, so it's especially relevant for border crossing species, that where if you want to preserve the species and make sure that it exists in the future, there are several countries who have to act in unison to enable that they are saved. So far, 51 countries have signed, and here is the list of the countries. Maybe you can spot your own country in here. I see Greece here. Where is it? Norway is here, Poland, Switzerland, and there are even 4 African countries that have joined. And since Norway signed this convention in 1979 and ratified it in 1986, and Sweden signed it in 1979 and ratified it in 1983, they have agreed to preserve the brown bear population, or the brown bears that live in these 2 countries. In order to, sorry that I'm sneezing all the time. I'm a little bit have a cold. I hope that's not too disturbing. So in order to preserve or to make sure that the brown bears exist in the future, it's important to know certain things. First, we have to know how many bears are there in the area. Then we have to know where are they. We have to know how much room does one bear need in order to be able to say, okay, for how many bears can still live in this country, because of course now there are so many people in the area that that you have to balance the needs of the humans and the needs of the bears, and for that it's necessary to know how much room does one bear take. Then you need to know, okay, what's happening is, are the number of the bears, are they increasing or are they decreasing? If they are decreasing, for example, is there something we can do to stop the decrease of the bears? How many young are produced every year? That is important. That is an indicator of how healthy the population is. And how many males and females do you have? If you have a population of bears, so a group of bears that only consists of males or of females, you have a problem, because then there can't be any young and you can't have any population growth. So traditionally, this monitoring was carried out by, for example, reporting whenever somebody saw footsteps of a bear, for example. They reported this. Where did they see it? And then, you can see a meter here. They estimated how big the bear was, and they recorded this. Whenever there was damage that they thought was done by a bear, or whenever they suspected that a bear, for example, had eaten sheep. The problem with this is, of course, you never can be sure whether how many bears did you actually report. If you have on the morning you see a bear at one spot, and then in the afternoon you see a bear 50 kilometers further away, you cannot be sure. Is that the same bear? Is it 2 bears? So this method, these traditional monitoring methods, are fairly insecure, and they tend to overestimate the bears. So they tend to give a higher number of bears than actually are present. So since 2006, the monitoring of Sweden and Norway is based on non-invasive sampling and DNA. I've in my previous talk, I don't know how many of you attended the first talk I held. With DNA, you can have a certain identification of each bear, so you know exactly which bear it was that produced here this pile of bear dung. You have information on male-female proportions, because you can tell from the DNA whether this was a female or a male bear that pooped this piece of poop. You can, if you do this over a long time, you can estimate the home range, so the area the bear uses, movement patterns, and family tree. All this is possible with the help of DNA. And I know maybe some of you attended yesterday the talk from my colleague Alex about hair traps. This is also a very neat method where the bear climbs under here and leaves some hair in the wire, which we can also use. There is DNA in the hair, and we can also use as a source of DNA for this monitoring. The typical workflow is that you have sampling. So you sometimes, I'll talk about this later, we use this also when there was a bear illegally shot. So sometimes we get these blood samples, or sometimes a bear has to be felled. Yeah, I see somebody attended. Very nice. Somebody, if a bear goes too close to people, you get, even though bears are not hunted here in Norway and Sweden, usually if a bear gets too close to people, you can get a permission to get it shot because they are afraid that bears might get used to, for example, look for food in trash bins. And to avoid that bears become used to looking for food with humans, then typically these kind of bears are shot if they are not able to scare them away. And then we get samples from these shot bears as well. And then here, bear from fecal matter, from DNA from fecal matter, from poop, and here from the hair. Then we register the samples. We write down where the sample comes from, and at what day it was delivered, or at which day it was found in the field. Then we do a DNA analysis, and we identify the bear, and then we can put this data on a map to see where the bear was. Just a quick recap. This is the DNA double helix, and this is where the information is stored. These base pairs of thymine, a T, adenine, an A, guanine, G, and cytosine, C. And you have a long string of letters which codes for certain information, how we look like and how we behave sometimes. But what we are out here for monitoring is the non-coding area of DNA. That are parts of the DNA that do not code for proteins. They are just there. We are not exactly sure in many cases if they do anything. And one part of this is called microsatellite, one type of this non-coding area, or short tandem repeats. And you can see here's the the flanking region where you have this AT, GC, AT, and so on. This is the DNA code. So the letters of the bases which code for something. And then you have here this, you'd notice that there's just two. There's GC, 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 GC all the time. And one GC here, you can see this is one repeat unit. And the entire microsatellite is the entirety of all this repeated pattern. And then you can have, usually we use repeat motifs of two, so GC or CA or TA. GC is very common, but you can have sometimes a repetition of three base pairs that you have ATG, ATG, for example. And do I have? Yes, and I have a little test for you on the next page. Let's see if you can identify. So here we have the same as before. I just took away the markings, and here's the repeat area. How many repeats do you count? Are you able to count so quickly? You can give the answers A, B, C, or D, and see if you can spot how many repeat units are there. And you have to remember, one repeat unit, this includes this and this, because it's a double helix. So this here is one repeat unit. Maybe I made it as big as possible, so I hope you can all see that clearly enough. Yeah, somebody else wants to answer? I've gotten the answers from 1, 2, 3, 4, 5. That's definitely less than from the first quiz. So I know that there are at least 3 that can answer. Okay, I'm going to walk you through the solution. Yes, thank you. this is one repeat unit, right? So if we count the G's on one string, we get the answer basically, because this is one repeat unit. So if we count, there's 1 G, 2, 3, 4, 5, 6, 7, 8, 9, 10. So there are 10 repeat units in this microsatellite. So C would have been the right answer. So since microsatellites, or STRs as we call them for short, mutate very easily by dropping or adding a repeat unit, and then we get variations of length, and these variations are called allele. So here you have the original 10 repeats, and then you sometimes have the variation. There's 2 repeat units added, then you have 12 repeats, or 3 repeat units. 3 repeat units drop, and then you have a shorter version of this, a short allele of only 7 repeat units. And the neat thing is that there is a lot of variation, and we use a lot of these microsatellites in our research and in our monitoring. And as I said also in the DNA, and probably most of you know, you get every individual has 2 DNA copies, one from each parent. And each parent, of course, has their unique length pattern of microsatellite alleles. And this means that you have, for each individual, because there is so much variation, a unique pattern of allele length combination. So you can see this is individual 1 has this type, a long one and a short one. This one has a shorter one and a bit longer one. This one has 2 longer ones. And if you do this with a large number of markers, you get a unique pattern with which you can identify each individual. But how do we get to the DNA? Well, we have the DNA, and because the poop runs through the intestines, a little bit of the cells that line the intestines gets dislodged, and they end up on the surface of this little poop pile here. And we wash this off, or we take this off and put it in a tube. And then we do something that is called lysis, which breaks down. Here you can see there are cell, the surrounding of the cell, and the DNA is actually here in this little blue dot, the nucleus. And with lysis we break down all this and free the DNA. But then, of course, we have also other food stuff, for example, if we have DNA from the fecal samples, we have a lot of matter that we don't actually want. We have non-target DNA, and we have bacteria, and we have undigested food, and we need to get rid of all this. So what we do is we add something that is called DNA binding. We put the DNA in this. I try to make this. This is a little filter, a very small filter that sits here, very fine mesh, and we add a chemical that attaches itself to the DNA, so the DNA gets really long and big, and it cannot go through this filter. And then we put this or everything into the centrifuge, and by spinning it around very quickly, everything that we do not want that is loosely there in this liquid gets washed down through the filter, but because the DNA is so large and because of this binding agent, it remains in this filter. And then we wash it. We rinse it with additional chemicals that we suck through this filter. And then in the last step, we put on a elution buffer, it's called. That is another chemical that takes away this binding agent and makes the DNA so small again that it can slip through the holes in the filter. And then in the end, we have the clean DNA here that we can work with further. And then we have polymerase chain reaction. I talked about this very quickly only now because it is complicated. In a yeah, we can heat the DNA up in a special machine, and it's separate, and then we add something that is called a primer. That is here a piece of DNA that can attach itself to this flanking region, and then there is a color, a flag attached to this piece of DNA here, and then the DNA builds up in a special process this part here, and then we repeat this everything, and at the end we have only the target sequence, so the microsatellite here with this primer and this flag, this fluorescent flag. And with a special method called gel electrophoresis, we can visualize it. We put a ladder. This is a mixture of DNA fragments where we exactly know how long they are, how many base pairs they are, and our sample DNA. Then you attach energy, electricity, minus and plus pole, and then the DNA is dragged through the gel, and the shorter, this should be actually the other way around, the longer fragments are stopped quickly, whereas the shorter fragments are quicker, and they go quicker. So this actually should be 600 base pairs, and this should be 100 base pairs. I'm sorry. I didn't have my head on my shoulders when I did this. And then because we have the ladder with the known length of the fragments, and this is the sample DNA, we know, okay, these two microsatellites, there are, for example, here 200 and 250 base pairs long. And then we get this, we combine this information from several microsatellites, and then, as I showed you before, each individual has a unique combination of these lengths here. You can see here there are four individuals. And we store this information in a huge database. We have thousands of bears in this database now. And whenever we get a new bear, we compare the new sample with what we already have. So another short test for you. Here we have the bears that we have already recorded. And here you can see the length combination of the two alleles, one from the mother, one from the father, for each of the 3 microsatellite markers. So now we get a new sample. And this is the length pattern that we record. Can you now tell me which of the 3 bears, which of the 4 bears this here is? Just compare the length. We do this usually with 8 of these microsatellite markers. There is so much variation that 8 markers can easily identify each individual. So we know exactly which bear pooped in the woods when we get new samples. Yeah, so it's bear number 3, of course, here. This is this little fella here. So this is basically what we do. We have this database, we get in samples, we analyze it, and then we compare it with our database, and then we assign here an individual. Okay, this is bear 3. And since we record where the sample was selected, we get a huge database of where each of the individuals that are in the database have been. And we have been doing this since 2006. So now we come to the part, what do we actually do? And this is basically what we're doing. You can see here from the area where I'm from, I'm somewhere here, there is where I'm sitting right now. And most of the bears are really here. This is only male bears now. And here you can see, for example, they have names which are dependent on the area where they come from. So the bears, all of the bears here are called FE and then a number. FI stands for Finnmark. This is the name of the area where I'm from the commune. So you can see, if you look closely, that FI 189 has been recorded here. and then somebody has collected poop here. Let's take another one. Yeah, it's a little bit. Yeah, FI 187, for example. Here is one, and there is one from FI 187. So if we do this over a long period of time, we get a good idea where each bear lives. And then you also, sometimes you get a bear that comes in here. This is Russia, and here is Finland. You get a bear that you only record once that maybe has been traveling, coming from Russia and going here, swimming over the river, pooping here, and then going further, and then maybe making its home in on the Finnish side. And then since we have been doing this so long, we know, for example, in this case, we have 27 male bears that we have recorded in total, and 15 of these we have not previously found. So these may be young bears that were not born before. It may be that they are so elusive that we didn't find the poop samples before, or it may be bears that just traveled through and that we therefore didn't find before. Also, we sometimes use this. This is a real case. Milieu Krim is here. That's an online newspaper. Milieu is environment, and Krim is crime, of course. So it's environmental crime, and this has been recorded there. There was that happened actually 2009, I think. I'm not quite sure, but some time ago. Somebody heard shots in the forest, although nobody was allowed to hunt there. So people grew suspicious, and when they went into the forest the next day, they found blood there on the ground. And they took the evidence from the blood, and they suspected that somebody had been hunting illegally bears. As I said, this is not allowed. You need either a permission or the official people. If there is a bear that has been creating problems, then there are official people who can shoot the bear. So this blood evidence was sent to our lab, and we analyzed it, and we were able to identify it based on our data bank on an as the individual HE27. And then police did their job, and they found some suspects, and they were a little bit stupid, those suspects, because they had the gun still there, and on the gun there was some blood. So they took samples. The police took samples from the blood. The men, of course, denied, no, we didn't do anything. But the police took samples from the blood that was found in the gun and sent the samples to us, and we analyzed it, and we could say that, oh yeah, this is the same bear. So these men were guilty of illegally killing a bear, and we, with the help of our database, could help convict these guys. Then, as I said before, if you remember, Norway does not estimate how large the population is. We only know that we have recorded at least 136 bears last year. But in the area here up in Norway where I live, we have done an estimate for the size of the actual population. So really how many bears are in this area. And you can see here the number of samples for each year. So as I said, we started 2005. Collecting samples, national monitoring started 2006. So we have 5 years from 2005 to 2010. This is a paper that a study that I performed 5 years ago. This was the number of identified samples for each year. And then based on how many bears were found, how many times, you can do a complicated statistical analysis and can estimate, okay, how many individuals are there actually. You can see, so for example here, from this year here, we have 137 samples. Each individual, there was 31 individuals, and each individual was observed, or the number of poops from each individual was about 4.42. And this number was used to estimate, okay, in this year there's two different methods. This year there were about 33 to 43 bears in the area. And you can see that sometimes it differs. For example, here we have 68 samples, 27 individuals. Each individual was observed about 2.5 times, and this method says, okay, there were about 36 to 39 individuals in this area. The numbers here say it's this has to do with the uncertainty of this method. Nothing is foolproof in statistical analysis. So on average, throughout the years, we can say there are about 40 to 45, 50 individuals that live here in this area. And then because we know about how large the areas they occupy, we can take this number and estimate how many bears live on 1,000 square kilometers. This 1,000 square kilometers is like a standard size that one takes. And these buff 15 and buff 7.5, that means we put on each sample, we put something round around it, and then we said, well, if they live on this area, there are about 11 bears per 1,000 square kilometers, or there are about 13 bears per square kilometers, or 5. So it's a bit complicated, but you can see that based on these samples, you can know already quite a lot about these bears. And then, as I said, that is also really neat. This is recorded. It's a little bit small, but this is from a bear that was caught over several years. And you can see that, you know, it walks around here. We have recorded it 2008 the first time, and then it was here and there, and then it went up there, and then it didn't like it there, and whoop, went all the way to the south again until it apparently made its home here in this area. And then we can also say with the population structure, I just briefly explain that we can see, based on the similarity of the DNA, how well they are connected. So how often a male bear, for example, goes in this area and has a baby with a female from this area. I will not go into this any further, but this shows that the population in Scandinavia, they don't have the bears that live in this area. This is the females and this is the males. They do not speak, well, they don't travel that much to the other areas. So they are, you know, like their own little village in a way. And this helps with the conservation also, with the that we know where the bears travel and how much they are in contact with each other. I'm going to talk in September, I think, or August. I'm not quite sure, but I'm gonna have a talk that looks at this issue of female and male bears. So stay tuned for that one if you're interested in hearing more about the bears in Scandinavia. Yeah, and this was it. Better with my time this time than the last time. So if you have any questions, please put them in the chat, and I will try to answer them.